Well, thank you to Susan for organizing this symposium and for the invitation to speak today. I'm going to talk about a different aspect of rabies in my presentation, which is to understand how it spreads and persists in a wildlife reservoir host, and specifically looking at vampire bats in Latin America. And the work that I'll talk about today has been spearheaded by Daniel Stryker, so I'm going to be up front in acknowledging that. And he started out as a PhD student in my lab and was later a postdoc and is now a fellow at the University of Glasgow. And there's many other people who've contributed to the work I'll tell you about today, including folks at the University of Michigan, at the CDC, and also many collaborators in Peru. So the results I'll present later in my talk today were originally motivated by a desire to understand how human activities influence the emergence of infectious disease, especially zoonotic infectious diseases. And there's a lot of ways that people unintentionally affect the emergence of zoonotic infectious diseases through activities like agriculture and urbanization, which were instrumental in the emergence of Hendra virus in Australia. Through the wildlife trade, which was implicated in the emergence of SARS coronavirus, and through deforestation and hunting, which have been linked to outbreaks of Ebola virus in humans and great apes. So all these are unintentional effects of human activities. There's also ways that people intentionally affect wildlife diseases by engaging in activities like culling and vaccination to directly control the pathogen within their wildlife reservoir sources. And we take a look at human activities and infectious disease emergence. It's really important to point out that there's a great need that still remains to better understand and predict disease dynamics within the wildlife reservoir hosts. And this is essential if we want to develop effective control strategies for minimizing exposures of humans and domesticated animals. And it's also essential if we want to be able to predict how future human activities are going to result in changing disease risk patterns. Now, in this context, bats have gotten a lot of attention, both from scientists and from the media, in terms of their role in harboring pathogens that seem to be particularly harmful for people and domesticated animals. So there are cases of pathogens like Ebola and Hendra virus and SARS coronavirus that have been linked to wild bat populations. And bats themselves have also been susceptible to devastating diseases, most notably white nose syndrome, which has killed over 5 million bats in North America alone and has caused local extinctions of several bat species. So there remains a great need then to better understand disease processes within bat populations to mitigate disease risks both for bats and for other animals that are susceptible to bat pathogens. Now, with that goal in mind, one system that's very tractable to study and that has provided some really interesting findings to date involves vampire bats and their associated rabies viruses in Latin America. So vampire bats occur throughout Central and South America, as indicated by the yellow shaded areas in this map. They can transmit rabies virus, which is a generalist pathogen, and the feeding ecology of the bats. So they feed on blood, and this provides a great mode of transmission for rabies virus as a generalist pathogen to infect humans and domesticated animal species. So every single night in Latin America, there are literally hundreds of thousands of vampire bats biting hundreds of thousands of animals. These bats bite livestock at night while they're resting or sleeping, so they will nick the skin and then lap the blood, which is a great avenue for transmission of rabies virus. And there are also many cases, many reports of humans being bitten by vampire bats, as indicated by this child with a vampire bat bite on the scalp. Now, some other aspects of vampire bats that make them suitable for study for host pathogen investigation are that they occur colonially, so the colonies range in size from about 20 to several hundred bats, so they're pretty tractable to study. The colonies are relatively sedentary, so it's possible to set up traps like this harp trap outside or at the exit or entrance to their caves and roosting trees, and it's fairly straightforward to collect blood samples and tissue and fecal samples from these bats and put bands on them for capture, mark, recapture analysis. Now, the vampire bat rabies system is also a system that's been really heavily affected by human activities. And one of those activities has been land use change for livestock rearing. So Latin America has experienced a massive amount of deforestation and conversion of lands to for agricultural purposes over the past 50 years or so. And for most species, this would be bad news. But for a species that feeds on mammal blood, like vampire bats, cutting down a forest and replacing it with a field full of cattle is like putting out a platter of food. And so there's been a lot of speculation that this land use change, that vampire bats have actually benefited from this land use change, conversion of forests to agriculture, and that this has resulted in both geographic range expansions of vampire bats and increases in their population growth. Now, given that we're potentially facing growing populations of a major infectious disease reservoir, it's not surprising that people are engaging in activities to lower bat populations or to try to lower contacts between bats and humans and livestock. And one way that they do this is by directly killing them, so by entering caves and either catching bats with nets and killing them, or setting fires in caves, or setting off explosives in caves. And people also kill bats using an indirect method where they use a vampiricide paste or a toxic paste and smear the paste on the fur of a bat that they catch. And then when the bat returns to its roost site, that paste is groomed off by other members in the colony, which then ingests this anticoagulant. Often warfarin is used in the paste, and then it kills the bats that groom the paste off of the target bat. Now, we can imagine two pretty drastic effects of these human activities of livestock rearing and of culling of bats on rabies viruses. So if we think first about livestock rearing, this is expected to provide more food for the bats, which could increase bat populations, could also allow for greater connectivity over space between bats from different colonies, and collectively this livestock rearing is expected to increase the transmission of rabies in vampire bats. When we think about the effects of culling, it's a little less clear how those are expected to affect rabies in vampire bats. So on the one hand, culling could reduce bat population sizes, and if virus transmission increases with bat population density, then anything that lowers bat numbers would be expected to lower viral transmission. Now, on the other hand, there could be some demographic and behavioral changes that would have the opposite effect. So culling could actually release bats from density-dependent control processes and increase birth rates, which recruits more susceptibles into the population and increases transmission. And we know from studies of other culling efforts for wildlife reservoirs like badgers that are culled to control bovine tuberculosis in the UK, that culling can lead to behavioral shifts where it increases the dispersal over space of animals and thereby also increases transmission. So today I want to show you some findings that touch on how these contrasting human activities of livestock rearing and culling affect rabies transmission in vampire bats and the risk of transmission to people and to livestock that encounter them. And in general, we've been going after this question in several different ways. So we have a long-term 7-year running field study that engages in surveillance of rabies virus in vampire bats in Peru. We've collaborated with people to develop some mathematical models to better help us understand the transmission dynamics of rabies and the impacts of control. And then also engaged in some genetic studies looking at host and virus population genetics to infer population structure and dispersal of the virus and the bats over space. So the first part of this work that I want to tell you about stems from a study that was launched by Daniel Stryker in 2007 through a collaboration between several different units in the United States and Peru. And this field work was also initially funded by a 2-year grant, collaborative CDC-UGA seed grant. So this involves monitoring 26 sites spread across 7 different departments in Peru, with each department indicated by a different color, and the site locations are indicated by the white dots here. And at each of these sites, bats are captured. I've got a picture here. So at each of these sites, bats are captured and they're tagged and samples are collected from them. And then those samples, the blood samples, are transported to the CDC, where we detect rabies antibodies using a virus neutralization test. Now, the sampling design allowed us to look at patterns of rabies in individuals and colonies over time, and I want to show you what some of those results look like in this slide here. So in this figure, each of the sites is indicated by a dashed line going to one of these 7 departments. And there's little tiny pies here which show the percent of bats that are seropositive for rabies. And the pies will get bigger in just a minute, so I know they're really hard to see. And in each pie, the white part, or the white slice of the pie, indicates the fraction of bats that are seropositive for rabies at a given site. The circles connect all of the sites that were sampled within a given year, and then the dashed lines are going to connect individual sites across years. And so if we move forward in time, so this was 2007 initially, and then we've got 2008, 2009, and 2010, what emerges is a picture that across all sites and most times, there are generally at least some seropositive bats, and there's not a huge amount of variation in antibody prevalence from site to site or year to year. So on average, about 10.5% of bats are seropositive at any given place and time. Now, because these antibody titers decline in the bats after a few months, we take this as evidence of fairly recent exposure if an animal is seropositive. So this is suggesting that rabies is around and it's persisting at fairly low levels in these bat populations over time. Now, we've also done analyses to better understand what factors do drive variation in infection and exposure among bats and among colonies. And so we've tested a number of colony-level predictors, including the past history of culling of the colony, the number of bats in the colony, and estimates of livestock density around each colony, and then individual-level predictors, including bat age, sex, and reproductive status. And in a nutshell, these multivariate analyses provided support for only 2 factors in affecting fine-scale variation in rabies antibody prevalence among these vampire bats. So culling history and bat age. And I'm going to tell you a little bit more about that in just a minute, but I want to also point out that we were surprised that neither colony size nor livestock density explained significant variation in rabies exposure among these bats, which the failure of these to explain that probably indicates that rabies transmission in these bats is not following what we would call a density-dependent process, whereby the probability of transmission increases with increasing density of the bats. So this suggests that is not an important component to rabies transmission in this system. Now, in thinking about the age effects, that's what I've shown here on the left panel of this graph. And so this shows seroprevalence over 3 different age classes: juveniles, subadults, and adults. And seroprevalence is higher in juveniles and subadults relative to adult bats. And this suggests, this is consistent with other work in bats, and it suggests that younger animals might either be exposed more often than adults, or that adults, after being exposed several times early in life, retain some protective low level of immunity. Now, when we look at the culling results, these were a little bit surprising. Maybe you would think they're even disturbing from a control perspective, because what they show is that rather than reducing rabies virus exposures, culling either had no effect or actually had a positive effect on seroprevalence in the bats. So bats that were from colonies that had never been culled had lower seroprevalence than bats from colonies that were intermittently culled or that were regularly culled. And so this suggests that culling is actually having the opposite, or might be having the opposite of the intended effect. And understanding what's causing this effect is really important, because it could inform better efforts to control rabies in bats in the future. So in terms of what's going on with culling, there are several reasons why culling might not be working well. One of those reasons is that the vampiricide might be disproportionately killing adults that had been previously exposed and are now immune, because those adults are more likely to engage in grooming activities with other adults. So the adults in the colony that might be offering herd immunity to the colony might be the ones that are getting knocked out by the culling, especially through the vampiricide paste, leaving behind susceptible juveniles that could better support rabies transmission. And some support for this comes from other data that suggests that the fraction of juvenile bats in colonies that are culled is higher than the fraction of juveniles in unculled colonies. Now, another idea is that rabies transmission, again, might not be related to bat density, and that's something that I alluded to earlier, which would suggest that culling should just simply have no effect on rabies transmission in the bat colonies. And a third idea is, as was seen in some other wildlife systems, like in badgers that were proactively and reactively culled to control bovine TB in the UK, culling might actually increase dispersal of these animals over space through several possible mechanisms, including if a colony is culled, that might leave a vacuum where there's more resources relative to the number of bats. So now more bats can come in to these culled colonies because there's more space or room for them there. It could also be that culling isn't inherently flawed, but it's just not being implemented well. And there's a better need to coordinate culling activities over space and time to make them maximally effective. So to really get at this idea then of what's happening, how can we explain the observed dynamics of rabies and vampire bats in Peru and these effects of culling, especially on rabies exposures? We first need to better understand what drives the transmission of this virus in bats and ask, how does this presumably lethal virus persist in wild bats? And to do this, we've developed a modeling framework to track rabies transmission within bat colonies. And essentially, this is a variant on an SEIR model where bats transition between an initially susceptible class to an exposed class upon encountering the virus. And then, after being exposed to the virus, they either move into an infectious class, which starts out being asymptomatic and then progresses to a rabid, lethal state, or after being exposed, they might alternatively transfer to this T class, which stands for temporary immunity. So they might experience sort of a subclinical immunizing exposure that never leads to an infectious and rabid state. And so the two states here in white would show up as seronegative in the field, and the states shown in black would show up as seropositive in the field. And then just to kind of tell you a little bit more about how this model works, so, and some of the parameters that are important here. So we have movement from susceptible to exposed to infected classes or to this temporary immune state. And so this parameter alpha defines the probability of advancing to a lethal infection. So bats move from an exposed to an infected state with probability alpha, or with probability 1 minus alpha, they move to this T, temporarily immune state. And then bats that move to this infectious state then ultimately become rabid and die. And there's a couple parameters that we know or that we can estimate from prior lab and field studies of rabies, and there's some important parameters that we don't know. And the first important parameter that we don't know is this alpha parameter, the probability of advancing to a lethal infection. And the other parameters that we don't know are the ones that control the transmission of the virus within and between bat colonies. And so we assume that this probability of exposure really depends on an underlying transmission process, which depends on transmission within bat colonies, indicated by these two terms here that have the parameter beta, which would be within colony transmission, and then the separate transmission parameter phi, which controls between colonies exposures and transmission. So we can think of phi as sort of picking up on the signal of movement of bats between colonies, or how often bats encounter other bats from different colonies, and how often that contributes to exposures to rabies virus. Now, This modeling work was spearheaded by Pej Rohani and Julie Blackwood at the University of Michigan. And to estimate these two key unknown parameters, alpha and phi, they applied a technique that's called particle filtering to the field serology time series data that I showed you earlier. So they used these time series data from the field, and I'm not going to go into the details of how this parameterization was done, but the goal was to estimate both the immigration rate, or phi, which is shown here along the x-axis, and the lethal infection probability, or alpha, which is shown here on the y-axis, and to basically ask which combinations of these two parameters best fit the observed seroprevalence data that we have in the field. And so the colors here show the likelihood of these parameters with increasingly darker colors indicating better likelihoods or a better fit between the model output and the observed field data. And the white star here indicates the maximum likelihood estimate of both of these parameters combined. And you can see that this falls out at quite a high level of intercolony transmission and a fairly low level for the probability of immunizing exposure, lethal exposure. So this suggests that only about 10% of the bats that become exposed to rabies ultimately go on to or advance to an infectious and lethal rabid state. So the bottom line here then from this exercise is that it suggests that frequent movement or immigration of infected bats between colonies with a low probability of lethal infection are essential for explaining the observed persistence and dynamics of rabies in the field data. Now, if we take a look at the predicted model dynamics in this region of space that represents the maximum likelihood estimates of the parameters, this is what we see. So this is the predicted seroprevalence over time. And this matches pretty closely the field data where seroprevalence bounces around between low values here between about 2% and maybe 15 to 20%, but averages around 10.5%, which is almost a perfect match to the field data. And then the colors here I know are a little bit hard to see, but the pink color represents the proportion of seropositive bats at any given time that are immune, and the blue indicates the proportion of seropositive bats at any given time that are infected and infectious. And this suggests that a very, very low percentage of bats in a colony at any given time are actually infectious, and that this is likely due to movements or incoming virus from other colonies, which then trigger really tiny outbreaks of rabies within these bat colonies. So this black line then is jumping up and down when these blue cases appear and lead to new exposures. So given that we're reasonably convinced that rabies is persisting through virus movement between bat colonies, we have a simplified model that kind of does a decent job of describing the field dynamics. We can take a look at how this model predicts different types of culling might affect rabies persistence in the bats. And maybe this can tell us something about why culling might not be working so well. So to remind you, one possible reason why culling might fail to control rabies in the bats is because we're removing the previously exposed and immune adults that confer herd immunity to a colony. And so we can track that through this. I'm going to show you some different colored lines by sort of varying the degree to which culling is discriminate versus indiscriminate within a bat colony. And so when the lines are red, that refers to disproportionate culling of older immune bats by that vampiricide paste. The blue lines refer to indiscriminate culling. There's an equal probability of taking out a bat from any age or infection class. And so we've used this model to look at how seroprevalence is expected to respond to culling activities. And this is a blow up, this inset is a blow up of the lines right here in this tiny part of the graph. And the first thing that you might notice, so here the black line is the no cull scenario, and then the different colored lines refer to increasingly discriminate culling. And probably the first thing that you notice is that it doesn't look like culling is having any effect at all on predicted seroprevalence in the bats. So there's almost no difference between all of these different colored lines. They're almost lying right on top of each other. The other thing that you might notice is that as we move, so this is just showing how culling performs across different values of phi, the immigration rate or movement between bat colonies that can lead to virus transmission. And what you notice is that this has a huge effect unobserved seroprevalence in the bats. And even tiny increases in the degree to which bats move between colonies can lead to huge increases in seroprevalence or exposures among the bats. And so this is a little bit alarming because it suggests that factors that increase bat dispersal, such as culling, which might be one of those factors, can actually make rabies a lot harder to control rather than making it easier to control. So I want to shift gears for a minute and think about what's happening spatially with the bats in terms of their movement across the landscape and the movement of rabies viruses. And to do this, Daniel Stryker and several other people, he's collaborated with several other people in Peru and at the CDC and at my lab to look at sequences of the virus and to look at genetic data in the bats to try to infer patterns of genetic substructuring and movement or gene flow over space. And so one way that this has been done is to capitalize on the large number of livestock cases of rabies that occur in Peru every year. And so this graph shows the number of monthly rabies outbreaks in livestock at a farm level from 2002 to 2012 in Peru, and this map shows the locations of these rabies outbreaks. And so in many cases, tissue samples from livestock that were affected by these outbreaks have been archived by the Ministry of Agriculture in Peru. And so our project's been collecting and sequencing virus from these samples, and that's some work that's been done at the CDC. Now, because transmission between livestock, so from cow to cow, is very rare, these data offer a window to pick up a signal of what's happening to the genetics of the virus within the bat populations. So genetic analysis was done of the nucleoprotein gene for rabies, and what this showed was evidence for 3 geographically structured clades of rabies virus in Peru. And so these are indicated as L1, L2, and L3, and this map of Peru shows the location where samples from these different clades came from. So L2 and L3 in the blue and green came mainly from the southern part of Peru, whereas L1 came from more northern regions in Peru throughout the Amazon and parts of the Andes. And so this suggests then that there is some spatial substructuring of the virus in space, but not to a large degree. And this structure seems especially curious because it provides a really poor match to vampire bat population structure that was derived from some mitochondrial gene sequencing that was done on vampire bats throughout the country. So bat tissue samples were collected from bats at 30 different colony sites, and this figure shows the location of those colonies where the tissue samples were collected. And then the different pie charts show the distribution of cytochrome B haplotypes in the bats collected for each colony. So the size of the pie corresponds to how many bats were sampled in a given place, and then the slices of the pie show the relative frequencies of different mitochondrial DNA haplotypes at bats within each site. And what this shows is that there's a huge amount of substructuring in the bat populations using the mitochondrial sequencing. So almost every single colony, or at least colonies from different regions, are genetically distinct from one another. And usually when we would compare the genetic structuring of a virus to the genetic structuring of the host, we would see more genetic structure within the virus because it's evolving so much faster than the host and it can pick up finer scale changes. And so here we see the exact opposite pattern. It seems like the virus is not respecting the genetic boundaries that are defined by host population structure. And so the question that emerges then is, how does rabies virus spread so effectively over space if the bats are showing so much evidence for spatial substructuring or sort of limited movement? Now, another view on this issue was provided by some additional genetic work that was done on 9 microsatellites using the same bat tissue samples that were shown in the previous slide. And what this showed was that there's a lot less spatial structuring in microsatellites, so these are nuclear genes, than in the mitochondrial DNA. And so a structure analysis actually picked up only 2 clades using microsatellite markers within the vampire bats, indicated by blue and by orange. And this, if we take a look at the map, matches the virus population structure much more closely. So here, the regions are color-coded as to the sites from which the bat samples came from in the blue and the orange groups. And the dots, again, show the different locations of virus DNA that was obtained, and the different clades here, the blue clade and the orange clade of the virus. And so this matches much more closely than the match that we saw with the bat mitochondrial DNA. Now, it's important, so a question is, what's going on here? And one possible explanation is that microsatellites are from nuclear DNA that's biparentally inherited, and so it reflects both male and female dispersal or movement across the landscape. But mitochondrial DNA is maternally transmitted, and so that would reflect more of what's happening with the females, but misses out on what's happening with the males. And so this could point to male bias dispersal in this species. So this disconnect between mitochondrial and microsatellite substructuring has been seen in other species too, and it's commonly thought to infer male bias dispersal, and so that might be what's happening in the vampire bats. An alternative explanation for the disconnect between the two different types of markers in the bats could be that because microsatellites are thought to evolve faster than mitochondrial DNA markers, it could be that they're picking up more recent changes. And it might be that historically, a long time ago, bats were a lot less well connected. But with deforestation and livestock rearing and human activities, it's increased the connectivity between the bats. And we're not seeing that in the mitochondrial markers because they evolve slower than the microsatellites. But if we do think about that first explanation of male bias dispersal, then these results could suggest that it's the males and perhaps the juvenile males that are largely responsible for moving rabies around in space, and it could point to a new target for possibly controlling rabies in this wildlife reservoir by targeting those juvenile males with control efforts. So just to sum things up then, in general, these results showed that rabies tends to persist endemically over time at a regional scale in Peru. And the modeling work indicates that between colony movement of the virus is perhaps the key to explaining the field patterns of seroprevalence. Both field and modeling studies suggest that culling is ineffective or likely to be very ineffective in controlling vampire bat rabies, especially as it's currently implemented, although it's possible that better coordination of the control spatially and temporally, better coordination of that culling might be more productive. And then in terms of the genetic work, the contrasting signatures of population structure from the two different bat markers point to key host groups for pathogen spread, in this case, namely males and perhaps juvenile males, which could play a disproportionate role in moving rabies virus around in space and between bat colonies. So I'll end again by acknowledging Daniel Stryker, who again spearheaded a lot of this work, and many of the other people who contributed to this study at the University of Georgia, at the CDC, and other locations, and then the funding sources that supported this work. And thanks very much for your attention.
We have time for questions.
Okay.
Did you look at all into the species of deer that is living symbiotically with the rabies that's being transmitted with the vampire bites consistently?
So we haven't done any studies of serology in any other wildlife species as part of this particular project. So I don't know the answer to that question, but yeah, I'd love to know more about it.
And the other thing I had a question, when I looked into why bats are carrying these more vicious viruses for us, what I learned is that when they learned to take flight, they had to develop an incredible system for handling oxidative damage to be able to free radical. And that's why none of these viruses bother them. And when I would see rabies expressed in bat colonies, like in Africa, it coordinated with a drought that year that killed the insects that they ate. And the mother bats were at peak lactation trying to feed and then they would actually express. So it seems to me that with any of these bat populations, what you're going to see with the expression of rabies is when they're stressed and they don't have food, or they're at peak lactation, or they've had some kind of environmental stressor on them that would allow the rabies to even be a problem.
That is a great idea. And that ties in with some work that another grad student in my lab is hoping to pursue, to look at the ties between the resources on the landscape and the degree to which bats are nutritionally stressed and stressed in other ways. So thanks for pointing that out.